Yes, Megha ma'am, please start.
Yes, sir. Good afternoon to one and all. We are back again with a very wonderful session for today. And today we are having Dr. Ranjan Kaushal Tirva sir from Sikkim. So good afternoon to all. And we are happy that we have again come back with the topic probiotics, where we still have a lot of concepts that can be covered, and we are going to see on what exactly is still needed to be covered. I welcome our today's speaker, Dr. Ranjan Kaushal Tirva sir. He is working presently as an assistant professor in Department of Microbiology, Nar Mahadur Bhandari Government College, Sikkim. Sir is also he has also received National Fellowship Award, and sir has completed his MPhil and PhD degree under the guidance of Assistant Professor Dr. Buddhiman Tamang sir from Sikkim University, India. Sir has contributed almost 8 papers in national and international journals, and sir is currently working on 2 book chapters. We are happy to have for today's session. So the session will be handed over to you. Just a minute. Sir, I'll start sharing the screen.
Thank you very much, Megha ma'am. And I would like to firstly thank our president, sir, Dr. Desmukh, for giving me the opportunity. So, with taking the permission of our sir, I'd like to start today's topic, that is probiotics. Myself, I'm Dr. Ranjan Kaushal Tirva. Currently, I'm working as assistant professor, Nar Mahadur Bhandari Degree College. So let us start by, can you just steer? Yeah, okay. So let us start with lab or lactic acid bacteria. Well, lactic acid bacteria are fastidious bacteria. Basically, it requires a special media or condition to grow. Well, they are lactic acid, they are gram-positive, catalase negative, microaerophilic, non-spore forming bacteria. And basically, it helps in fermentation. Purely fermentative with specific requirement for nutrition, such as it requires carbohydrate, amino acids, peptides, fatty acids, ester, salt, nucleic acids, and vitamins for fermentation. And it is abundantly found in rich nutrient area, such as in fermented products, in plants, animals. Well, it's very interesting. Why fermentation? Well, fermentation is a technique. See, in Sikkim, most of the people prefer traditional fermented foods. Likewise, in India also, so many fermented products are there. Likewise, in Sikkimese community also, we prefer traditional fermented food. So what is the idea behind fermentation? Before, so olden days, even people from mainland were microbiologists. See, our traditional, when we go back to our tradition, without using the help of technology, fermentation technology, what are we doing is we are trying to increase the shelf life of the product, various product like various vegetables. Like people of Sikkim prefer gundruk. What is gundruk? It is a traditional fermented product. It is a green leafy vegetable which normally the harvest is done normally during summers, during winter. Sorry. So before in olden days there was no technology for preservation. So what they did was they sun dried it. What they did was they use traditional fermentation technology. See, the green leafy vegetable is now totally different. The texture, the aroma, the flavor, everything has changed. And because of the fermentation, the microorganism has, what it does is it has so many added metabolites. It has added so many health benefiting properties. So let's turn that thing into probiotics. So likewise, due to the properties of certain members of lactic acid bacteria, such as lactobacillus, which has been extensively studied as a probiotic microorganism. Because of these properties, even these days, people have started using in therapeutic or in in research as therapeutic use. Okay, so the whole idea of lactic acid bacteria is it is probiotic bacteria, and it has normally health benefiting properties. Can we just go to the other side slides? So what okay, probiotics, yeah, okay, yeah. So what are probiotics? Probiotics is basically living microorganism which, when we administer in adequate amount, will confer a health benefiting property to the host. Likewise, I already told you, like, these are the live microorganism which, when we take in adequate amount, will have health benefiting properties. Not only that, see, these bacteria are normal flora, part of normal flora. They reside in your alimentary canal. In an individual, वो क्या करता है? वो alimentary canal में रहता है. So what it does is, it does not allow the harmful bacteria or pathogenic bacteria to come and stay, or they do not allow. So it is a part of normal flora. So probiotic has been reported to be helpful in irritable bowel syndrome, inflammatory bowel disease, even in constipation, even antibiotic resistance. Maybe it has helped, and even etiquette diarrhea. Maybe it helps, even hypertension. And these days, mostly anti-cancerous properties has been established, and anti-diabetic properties has been established. Mega, yeah. Okay, so this is my PhD thesis actually. So the rationale of taking this lactic acid bacteria was behind the enzyme inhibitor drug. See, these days many people are into so many drugs, right? They have to take, because of our lifestyle. So many lifestyle disorder is very prevalent in these days. In Sikkim also, because of lifestyle changes, so many lifestyle disorder is very prevalent here in Sikkim. So if we go for a medication, any meditation, be it any meditation, it will have long-term impact to your cardiovascular diseases, or it might have impact on your renal or kidney in a long run. So what we are trying to do is, with the help of this bacteria, probiotic bacteria, we are trying to get an inhibitor from there against alpha amylase inhibitor. So the enzyme inhibitor drug prescribed to inhibit alpha amylase and alpha glucosidase for the treatment of diabetes are not free from side effects. Therefore, an alternative and complementary method for management of postprandial hyperglycemia and diabetes is necessary. So the whole idea of this study was to curb diabetes or postprandial hyperglycemia using inhibitor from lactic acid bacteria. Before coming to lactic acid bacteria, I want to stress on postprandial hyperglycemia and diabetes. So diabetes type 2 is very common in India and also in Sikkim. Sikkim ranks among the first in terms of prevalence. So many people suffer from diabetes because of lifetime changes and so many other things. Like diabetes type 2 is a multifactorial disorder because of stress, because of heredity, because of lifestyle, because of sedentary lifestyle, because of your no physical activity, because of obesity, and because of postprandial hyperglycemia. One can have this diabetes type 2. And if we are to see the age group, late 30s, I mean early 30s or late 20s, people suffer from diabetes type 2. right? So what is postprandial hyperglycemia? Postprandial hyperglycemia is a risk factor associated with diabetes type 2, right? In postprandial hyperglycemia, there's a condition, well, because of the enzymatic activity of alpha amylase and alpha glucosidase, the sugar level in the blood goes higher. It shoots up like anything. It lasts for another 2 hours. Okay. But initially, no impact is seen. But later, the window period of postprandial hyperglycemia is 10 to 12 years. Then the person will switch to diabetes type 2, right? So that is the thing. So if somewhere we manage postprandial hyperglycemia, somehow we will somehow the people will not suffer from diabetes type 2. The whole idea was to curb stop postprandial hyperglycemia by using the inhibitor from lactic acid bacteria, which is a probiotic bacteria. I hope everybody is getting it.
Yes, sir.
Okay. Okay, sorry. Okay, I'll be later. Okay, so the the objective of our study was to isolate lactic acid bacteria from various sources like milk, traditional fermented products of Sikkim. And once we have this bacteria, we screen this bacteria against digestive enzymes, that is alpha amylase. Right? We are supposed to look for inhibitor against alpha amylase. And once we have this bacteria which is showing alpha amylase inhibitory property, we went and we looked for the other probiotic properties as well, right? And then we were much interested in identifying this bacteria using both the method, the conventional method and the modern method, like phenotyping. We studied the phenotypic, we did the phenotypic identification, and then we use the modern technology like genotypic identification was also done, right? Yeah, and sample was collected from various locations of Sikkim, like various samples were collected from different parts like east, west, north, west, everywhere we went, and we collected local samples, like we collected milk samples, dahi, churpi, and we collected dahi, and we collected gundruk. It's all local traditional fermented foods. Can we go to next slide, please? Yeah, and the traditional fermented foods of Sikkim, basically it's bamboo shoot. It's we call it meso. It's consumed by all the people, and fermented leafy vegetable that is called gundruk. Initially, it is leafy vegetable. Once we sun dry it and we ferment it, and then later the aroma, texture, everything is changed. Same goes with the fermented radish that is called sinki, and fermented soya bean that is kinema, we call it, is similar to natto, kimchi, something like that. Okay, and marcha, it is a amylolytic starter. And from milk also, even from milk and milk product, we use local milk, cow milk. And churpi is precipitation of this milk, coagulated milk. And we also go for mahi, chach, it's called chach. And we also go for ghee. Ghee, it is butter. We collected yak butter and cow butter was done, and even human source sample was also considered for the study. Infant babies sample was collected, stool was collected from infant baby, and lactating mother was also considered for the study. And isolation was basically done using the standard method. We collect the sample and saline was used, and later serial dilution was followed by pour plating method. And then we make sure that our cultures were safe. We did the streaking, and pure cultures were there. once the pure cultures were there, we incubated for another 6 months under 80 degree. And then once we had this culture, more than 300-400 cultures were there. And then we started the screening. Okay, screening for what? Screening for alpha amylase inhibition by using star identity method. Okay, initially we tried it so many methods, but we found it very helpful for our study. So we stick to star identity method for screening against alpha amylase. So we took the culture that was 18 hours old up to 24 hours old. Culture was taken, and then centrifugation was done. And because our culture, because our bacteria, because our inhibitor is extracellular, so we use supernatant in our study, right? And then the culture was incubated in 50 mu protein alpha amylase that was provided by Sigma. We use Sigma standard culture was there, alpha amylase was there, and then we add another 40 mule supernatant to it, and then we incubate for another 10-15 minutes and see, and we add another 40 mule starch solution. Why? Because starch solution is your is your substrate for alpha amylase, right? And later the reaction was quenched using the iodine solution, and then later OD was measured. Inhibition percentage was calculated using the formula. For our study, acarbose, which is a known inhibitor, right? And copper sulfate is also a known inhibitor, was also used as a positive control, and MRS broth was used as negative control. And see, once this is done, we could get at least 10 isolates, which were showing very good result. And for production of alpha amylase inhibitory this alpha amylase inhibitor, what we did was we did the study at what time the activity is at the peak. So we want to know the bacteria, at least 9 bacteria were selected. For that bacteria, what we did was we studied the time graph, or we started from 0 to 96 hours. We, at interval of 8 hours, we took the reading. Okay, the selected isolates were cultured in 100 ml MRS broth in 250 ml culture flask under shaking condition at 37 degree, and then 250 ml of MRS broth in 250 ml was also, see, for this we have used MRS broth as control. And then activity was checked from 0 to 96 hours. At every 8 hours we used to go for this assay. We used to check the activities were assayed as per the method that is screen. We use the same method to screen the activities. And see, our culture, our inhibitor is a crude, okay, which is not in the pure form. But then also we wanted to check the stability of our culture. We wanted to know at what temperature the alpha amylase activity is at the peak. So we we made a 3 profile, like room temperature was considered, and then 8 degrees refrigerated 8 degrees were considered, and -20 was considered for the study. And then every week we used to check the activity. And then since the other probiotic attributes are also important for our study, right? So we did 2, 3, 4, 5 important profiles, like survival strategy in alimentary canal, whether our bacteria can withstand the acidic condition or bile condition in our stomach. So we mimic the thing artificially in our lab, and we try and see whether our bacteria can survive or not. The other test we did was hydrophobicity test, and then followed by hemolytic activity, and then antibiotic susceptibility, and auto aggregation techniques was also followed. See, so you must be aware of the fact, like prebiotics. So what are prebiotics? Prebiotics are basically are foods, okay, typically high fiber foods that act as a food for human microflora. So we wanted to see the utilization of this prebiotics, such as sorbitol, inulin, raffinose, oligofructose, xylitol, and mannitol. And even this was studied. So for phenotyping, we use Burgess manual for identification, and for DNA isolation, we use phenol chloroform method. And then we went for 16S sequencing. And then PCR was done, but we don't have the facility of 16S sequencing. It was outsourced. And then once we had this FASTA file, we submitted this our SSN number. All the 16S RNA gene sequence were submitted in gene bank, and then SSN numbers were also obtained. And we followed for phylogenetic maximum composite livelihood method was used using the Mega software. So two isolates were selected for whole genome sequencing, that is DMR 60 and DMR 17 and DMR 9, because they were showing very good results. So it was also outsourced because we don't have the facility out here. So DNA, basically the whole idea of this is DNA fragments are generated, and then thirdly, it is followed by assembly and then annotation. But analysis was done by me here itself. So basically, we use various tools to for analysis, like RAST was used for annotation, right? For plasmid fight, we have to remove duplicates, and then so many platforms were used, like plasmid fighters were also used. RAST was used. Can we go? Yeah. For plasmid finder were used. A Patrick platform was also used, and then we have this PGPA was also used for annotation, and then for RAS was also used for annotation. So this is the thing for Patrick platform. RAS was also used for annotation. Let's get back to result, okay? And these were the samples from various locations of Sikkim. Mostly milk dahi was there, and then this is the result. I just wanted to discuss the result. Can we go to result? Yeah. So this is the result for my study. As we all can see, in case of DMR 09, it's almost 90%. Okay, it's 90%, and DMR 15, which is showing 73, which is the lowest. Yeah, this was done in the triplicates. 5 datas were there and done in triplicates. Okay, and this is the screening method. The method that I did was using the ELISA plate reader plate, 96 well plate. We all can clearly see. We can see first plate, first 6 wells is dark, right? And the another 6 is not dark. Why? Because in the another 6 here you all can see, because here the reaction has occurred. Here your starch and your enzyme has, the reaction is there. Because the reaction is there, we cannot see the dark color. And in this case, this is MRS broth. MRS broth is there. Your alpha amylase is here, and then starch is here. So reaction, we are the whole the whole idea is the alpha amylase has degraded your starch. So reaction has occurred. In this case also, alpha amylase has degraded your substrate, that is starch, right? But see, in case of this thing, we cannot see the reaction because of the inhibitor. The reaction was not possible, right? Because of inhibitory activity, the reaction was not there. Yeah. Okay. We all can see the box plot showing the distribution pattern of alpha amylase activity of inhibitor from different isolates. Yes. So this is the result for production of alpha amylase activity. Here, DMR17 showed a very good result, and then 9. Yes. So, so this is the result for survival studies during orgo-gastric intestinal transit. All this bacteria, 9 isolate, survived the harsh condition of our. We were trying to mimic the alimentary canal, and all this bacteria could survive in a showed a very good result. And all this bacteria could survive the harsh condition. Yes. So this is the result for hydrophobicity test. So adherence ability of different lab isolates to hydrocarbons such as N-oxidine, N-octane, xylene, hexadecane, chloroform, and all this bacteria could survive this test also. Yeah, this is the tabular form of the result. Yeah. Even see, in case of hemolytic activity and gelatin hydrolysis, all this bacteria had a negative result, right? So for auto aggregation also, it showed a very good result, and all this bacteria could 16 being the highest. 16 showed very good result, DMR16. Yes. So this is the tabular form of the result. And even for the probiotic, all this bacteria could utilize all this probiotic, prebiotic, very properly. They showed very good result. This is the result for your crude stability. And minus 20, the bacteria where we had stored, the crude extract where we had stored in minus 20, it showed a very good result. So this is the identification on the basis of cell morphology and the convention method. Like these bacteria were basically rod in shape, and then these are like lactobacillus species. So this is the gel picture. So this is the gene accession number where we had submitted, and we have got this accession number also. All these bacteria are Plantarum. Can we go back once? Yeah. So interesting thing is we have DMR8. DMR8 is also a lactic acid bacteria. It is Plantarum only. The interesting thing is it has all the profile. Everything is there, but it is not showing the inhibitory activity. So we are using it as control. So this is the phylogenetic tree. Yeah, this is the profile of whole genome sequencing. We could get one plasmid from both the isolates, and RAST subsystem annotation showed it has 226 genes were corresponded to carbohydrate metabolism. Among them, 18 genes are responsible for lactic fermentation and 23 genes for mixed acid fermentation. So even pathways were studied, and EMP was there. And this is the figure pathway of lactic fermentation. I cannot show you properly. This is the subsystem. This is the photograph from RAST, and we can see the different cofactors, different biomolecules are there, like biomolecules like carbohydrate, how many carbohydrate genes are there, something like that. We could see everything is there. They in RAS they give everything in detail. So this is the circular representation of assembly complete genome. So I would like to discuss here. We had more than 300 lactic acid bacteria isolates from different sources, and we screened these bacteria against alpha amylase, and from there we could get, we somehow managed 7 isolates showing a very good result that had alpha amylase inhibitory properties. So these results were reproducible, not only once, not only twice. So many times we tried, it was reproducible. So we continued our studies like DMR 9, 12, 13, 14, 15, 16, and 17. And DMR 9 and 17 was considered further for whole genome sequencing. So the highest was in case of DMR 9, that is 90%. The alpha amylase inhibitor production corresponding to late exponential phase of growth for all the isolate. DMR 8 could not produce any inhibitory activity, as its growth characteristics and acidification of medium were similar to those alpha amylase positive isolates. Moreover, the MRS medium was also negative for alpha amylase inhibitory activity. This indicates the alpha amylase activity is not because of acidification, nothing to do with acidification of growth medium or due to the component of growth medium. The best selfie supernatant was preserved at -20. So for this survival studies, to elicit the full effect in host, we try to mimic the harsh condition. So most of the bacteria isolates from food samples are very sensitive to acidic condition of our stomach. As we are aware of the fact, our stomach is very acidic, and then it's very difficult for organism to grow. Likewise, but our organism could readily grow well in acidic condition because of presence of proton exclusion mechanism. So all the isolates show very good result in vitro stimulation of orogastric intestinal tract. Therefore, there was a good number of survival population log, that is 5.2 log CFU per ml, even at the lowest gastric pH, that is 1.8. We did that also, which was similar to respond to lactobacillus acidophilus under a similar condition. Probiotic organisms should also tolerate bile, right? And even our bacteria were bile tolerant. So for hydrophobicity test, the probiotic bacteria should somehow stay in GI, right? So for a short time, therefore, to attach the intestinal mucosa, the cell surface of probiotic microorganisms should be hydrophobic in nature. So all this isolates should moderate to high. And in case of this 16, DMR 16 is a very good result. It adhered to hydrocarbons such as N-hexadine, N-octane, followed by chloroform and xylene. So the partition of microorganism cell to hydrocarbon phase indicate that integration of cell surface to mucosa through van der Waals and electrostatic interaction rather than hydrophobic in nature of the cell. So our bacteria was negative against hemolytic activity and gelatin hydrolysis, which is a very good result actually. So antibiotic susceptibility also, all these bacteria were sensitive. This isolate need further investigation. We also wanted to further study this because all these bacteria were sensitive to the antibiotic that we used. And prebiotic, all these bacteria use this probiotic, especially sorbitol is one of the emerging probiotic component, although it's observed in the upper GI. And it's very slow, though a significant portion of sorbitol can reach to the colon to be utilized by colon bacteria. But this also has been utilized, properly. So all this for RAST, I mean the whole genome sequencing, alpha amylase inhibitor was selected, that is DML9 and DML17, and we could find so many enzymatic activity like fermentation was there, using so many pathways we could find in our study. Okay, I'll just conclude this lecture. Out of several lactic acid bacteria, it was initially 300 plus bacteria, but we stick to 7 isolates. Lactobacillus plantarum, isolated from homemade cow dahi, were able to produce alpha amylase inhibitor. This is the first report that describes, okay, the alpha amylase inhibitor from Sikkim formatted products. Though lactic acid bacteria can utilize some of the probiotic component, and it can be withstand the GI harsh condition also. Therefore, the lactic acid bacteria can be a potential probiotic candidate which can be used the control of postprandial hypoglycemia and management. So we just wanted to continue our study. So I would like to express my gratitude to supervisor, Dr. Buddhiman Tamang, who is an assistant professor in Department of Microbiology, Sikkim University. If you have any query, please get back to me. I'll be happy to help you. Thank you very much. If you have any query, please get back to me.
Sure, sir. So there is a question from Prakash, sir. Did you use standard probiotic culture for comparison with your native isolates?
No, we did not use, but the reagent, but okay, standard. We did not. We compared with the help of some papers. We did not use standard, ma'am.
Okay, sir. So my question is, whatever isolates you got, you were checking it for its alpha amylase inhibitors, and you even tried to check the antibiotic susceptibility.
Yes.
So if the organisms are susceptible to antibiotic, is it really efficient enough to use these organisms as probiotics?
No, actually, in my case, there is like very limited study. We have used very limited antibiotics. So we just wanted to continue further, and we just want, it's very initial stage, no? Our study is in very initial stage. So we wanted to continue, and whether we want to see genetically whether it is, we wanted to know whether gene has been transferred or not, something like that. We wanted to check for our study. The main focus was alpha amylase inhibitor activity, and these probiotic attributes will also help, no, to establish more authentic. So that's why we did this probiotic study.
Okay. So one more question: what percentage of microflora? Yes, sir. Someone is trying.
Yeah, I have a question. Yeah,
Yes,
did you establish the adequacy of the dose?
No, it is the study is it's at the early stage, and moreover we have used this crude extract. We wanted to purify this culture, and then we wanted to.
Your claim is probiotic, right?
Yes, sir.
The very definition of probiotic is, as you have said it in some of your slides, is beneficial organism added in adequate number causes host benefit.
Yes, sir. Yes, sir.
So you are speaking of host benefit. What about adequacy? Did you establish that? Hope you got on my question.
Yeah.
Yeah, Dr. Prakash Halami would be able to explain it more explicitly.
Yes.
Sir, good morning. Actually, we are working on the clinical trial of a Gundruk prepared in
exactly. Because unless you establish the adequacy, you can't determine the dosage.
Yes, sir.
And the commercial aspect of it would depend on whether 10 raised to 9 are adequate or 10 are adequate or 11 are adequate.
Tomorrow is the last date to submit the call for proposal under a DBT program.
Acha, you are submitting this, huh?
Yes, yeah. So basically, they are asking the any fermented food which we are talking
Yeah,
has
yeah.
clinically proven for their health benefits. And last week, in fact, I was
Obviously, unless you prove clinically, it's of no use.
yes, sir. Yes, sir. Anurag Krupadap, John's Medical College, Bangalore, and he said
Yeah.
that possibility, studying the fermented food for health benefit is their uniform consistency and the dosage is the
No,
question mark.
and yeah, and at another thing is the metabolite which is giving you that inhibitory effect. And why the hell you go for borrowing the organism, isolate the metabolite and administer it, which is more easy.
Exactly. We will propose your name as one of the potential referee of our project.
No, kindly don't do that. Maybe I'm not efficient or competent enough to. I mean, whatever has come to my mind, I'm just speaking out.
Exactly, it is very much valid question, and then there is
Yeah.
actually the provision to suggest the 5 referees expert in the field. So we will be very happy actually if
I'm not an expert. That is number one. Then I know less into this field because I had been always on sporulatic probiotics than on vegetative forms. So
Yeah,
I had not be the right person.
So sorry to interrupt. Are there any more questions for the session?
Yes, I have one question.
Sure,
This is
sir,
Dr.
please.
This is Dr. Krishan. Good afternoon to all of you. Sir, my question is that you got 2 isolate, DMR9 and 17.
Yes, sir.
And can you tell, sir, what is the origin of these culture? I mean to say from where you got these 2 isolates?
This is from cow dahi. Cow dahi. Origin is dahi. Local cow dahi.
Okay, and you got the whole genome sequencing of both these 2 isolates.
Yes, sir.
Sir, when we compare the AAI activity of these 2 cultures, then there is a substantial difference between the 2.
No, actually, these 2 cultures were showing very good inhibitory activity. So we consider for whole genome study. But at genetic level, we couldn't find any gene related to alpha amylase inhibitor.
Okay, that's my question. Is there any is there any gene copy number that
No. We couldn't find any gene that is related with alpha amylase activity. We tried so many times, but we couldn't get any gene related to alpha amylase activity, sir.
So, sir, in your opinion, what could be the reason behind this observation? Higher AAI activity without any gene.
See, the same bacteria, in case of DMR9, no activity was found. And we tried so many times, but no activity was found. And that's why we thought we will have to go for the whole genome sequencing to rectify genetically, but we couldn't. And it's like this study is very at the initial stage. We wanted to further continue this study actually, sir.
Okay, thank you, sir.
Okay.
Sir Ranjan, sir, there is one last question from Dr. Vinay. Which tests are there to prove that the cultures are probiotics?
The one that we did are the standard one actually for our bacteria.
So they are species specific.
Yes.
Okay, sir. So I guess there are no more questions for today's session. So here we are at the end of the session today. So I would like to thank Dr. Ranjan sir for giving his valuable time on Sunday and giving us another aspect of probiotics apart from milk and milk products. So we are very grateful to you, sir. Thank you for your time and the valuable knowledge you have helped us to gain on today's session.
Thank you very much.
I would also like to thank Dr. Deshmukh sir for providing the opportunity for conducting such awesome lectures for Sunday's session. And last but not the least, I'm also thankful towards all the members who have taken out time and shown their interest in the session and have asked wonderful questions regarding the topics. So thank you to one and all. Hope we'll have the same strength for the next session, where we'll be having the session on the provisions or the tests that are given by different organizations and what are the fee structures for that. So thank you, sir. Thank you to
very
one
much.
and all.
Thank you very much. Thank you, everyone.